Hi. So we are in the middle of this course, and this course was started with historical aspect of immunology, and then we learnt about the immune organ. Then we have learnt various immune cells, and then we have learnt about the cytokine molecules, which basically coordinate the cells and various component of immunity. And after that we have discussed about uh, there is a pattern recognition receptor. We have completed this pattern recognition receptor. We learnt in quite deep length. Okay, and today I will start one more component of uh, immunity, or more precisely innate immunity, that is complements. So, complement is kind of a junction between innate and adaptive immunity. Okay. This complement activation do need the support from adaptive immunity in terms of antibody. Okay. And you know that antibody is produced by the B cells, and B cells are adaptive immune cells. Okay. So, complement is also a component of innate immunity. It is a humoral component, it is a soluble component which is playing a very important role against microbial infection. Okay. Today I will begin or in this session I will begin the complement, and before beginning I would like to take you again in historical part, how it was discovered. Okay. So, if you remember my previous or initial sessions where I have discussed about the history. So, if you remember, then you probably remember the name Pfeiffer or Pfeiffer phenomena, which was discovered by Richard Pfeiffer, okay, around 1894. Okay. So, this work basically started, the initial observation was started with his work, and uh, later on this work was taken up by Jules Bordet, and he established that there is some factor which is playing very important role in bacteriolysis. So, let me explain the Pfeiffer's work. Okay. So, Pfeiffer basically working on guinea pig, and he was using Vibrio cholerae as a model bacteria for his experiment. Okay. So, what he has done, he has performed one simple experiment where he challenged the guinea pig, which is immunized with Vibrio cholerae. Okay. And he noticed that in this guinea pig, the when he exposed this guinea pig with Vibrio cholerae, which is already immunized with Vibrio cholera, okay, or you can consider that a guinea pig recovered from the Vibrio cholerae infection. So, then in that animal he injected in peritoneal cavity, he injected Vibrio, and then he noticed that this bacteria is very quickly disappear, or it will be very quickly lysed. Okay. So, this observation and he, along with this experiment, he performed another experiment. He also performed this experiment using non-immunized animal, and when he injected, please listen carefully, when he injected the non-immunized guinea pig with the serum which is taken from the immunized guinea pig, and the bacteria, the bacteriolysis was happened. Okay. So, when he performed the similar experiment in vitro condition, he was not able to show the bacteriolysis consistently. Okay. And finally, what he concluded out of all these experiments, that there are some phagocytic cells which is present in peritoneal cavity, and those cells basically cause the bacteriolysis. So, this work was till this point where this Pfeiffer has completed this work and concluded, his conclusion was it is some peritoneal cavity phagocytic cells which cause the bacteriolysis. Okay. Then what happened in around 1895, Jules Baudret basically initiated his experiment with his observation, Pfeiffer's observation, and he was basically working in the lab of Emil Metchnikoff. You remember the Metchnikoff who discovered the phagocytosis. Okay. So, he was working in his laboratory, and his project is to investigate this Pfeiffer phenomena, okay, extensively. And he performed a series of experiment using Vibrio and guinea pig, using antiserum and serum, and he finally concluded that there is some factor in the serum, okay, and this factor is partially play an important role in bacteriolysis. Okay. And if the serum is not kept properly, or if it is not fresh, if it is old, or if the serum is exposed to the temperature fluctuation, particularly if the serum temperature increase more than 55 degree, then this loses its bacteriolysis capacity. Okay. So, this was the beginning of the concept of complement. He very firmly showed that in fresh serum, besides antibody or besides a specific molecule, there is a molecule which is needed for the bacteriolysis, and this molecule is irrespective to the specificity. Okay. It means if the serum is old, which is received from the immunized animal, okay, then it will not show the lysis activity. Okay. But if it is replenished with fresh serum from non-immunized guinea pig, then that complement activity will be restored, or the activity of the serum will be restored irrespective of any other thing. Okay. So, this was a great experiment and great observation, and that result to the discovery of complement. Okay. So, this work was done by Jules Baudret. Beside this, he has performed various work during his lifetime, and he also discovered the assay which we call it as a complement fixation test or assay, which is used for the identification of a specific antibody or a specific antigen. When I say antigen, it means a particular microbial pathogen. Okay. So, in addition to these two very key discovery, he also discovered the causative agent of whooping cough. Probably you are aware that whooping cough is caused by Bordetella pertussis. Okay. And so, he is the discoverer of this Bordetella pertussis, which causes the whooping cough. For all this work, he received the Nobel Prize in 1919. So probably you might be aware that 1919 or 1918 was the pandemic time, the influenza. There was a pandemic all over the world. Okay, so at that time he received this Nobel Prize. Anyway, let's come back to the complement. So basically he was working in Metchnikoff laboratory at Institute Pasteur, and he after his experiment his observation was that there is some factor in the serum which is heat labile factor, and this heat labile factor is responsible for the bacteriolysis. Okay, and he also suggested that these complement proteins or these factors are present in zymosin form. Okay, so this zymosin form is inactive form, like a you know that there are several enzymes which are present in inactive form, and we call it as a zymosin form. Okay, or pro form, and this pro form get activated when there is some cleavage of this protein. Okay, so this was the observation. He also showed this phenomena in sheep, and the way I have explained, he have performed this experiment in using sheep antiserum, and he showed this if you heat the serum at particular temperature, that is 55 degree centigrade, then serum will lose its lytic activity. Okay, and lytic activity can be restored when this heated serum is mixed with fresh serum, which is irrespective of pre-immunized or immunized. Okay, so this was his all work, and finally this heat sensitive component is, he established that heat sensitive component is important for bacteriolysis, and this augmented activity of antibody is also observed by Paul Ehrlich. You know that he is very important in giving the side chain theory. Okay, so and since he observed that this antibody activity is augmented by these factor, he gave the name complement. So the complement word is given by him. Now let us look at the component of complement. So complement are basically a group of protein which is present in zymosin form in inactive form. Okay, and this is basically a protein or glycoprotein and synthesized mainly in the liver and other cell types, and 5% of serum globulin is the complement. Okay, circulated in inactive form in zymosin form, and there will be a cascade of signaling or proteolytic cleavage that makes it inactive to active form. Basically it cleaves the inhibitory fragment and expose its active site, and in that way this complement get activated. The outcome of complements are three major outcomes. One is I think told you several times in previous session. The first outcome is one of the outcome is the lysis of target cell. So when complement get activated, this will lyse the target cell. Okay. Another is opsonization. So opsonization is basically the target microbe will be coated by some group of molecules. Okay, and once it is coated, then phagocytic cells will readily phagocytose these microbe. So we call it as a opsonization. Okay, or you can call it as there will be a more quick phagocytosis. The third outcome of the complement activation is to induce inflammation. Okay, so some of the complement some of the complement part is if it is cleaved from the parental complement molecule, then that will act as a inflammatory substance. Okay, it induces inflammation. Okay, and you know that inflammation is a very well. I have discussed inflammation in quite big length in previous session. Okay, so this complement can also cause the inflammation. Okay. Now I will talk about the nomenclature of complement. It is it has some set of naming system. Okay, so all complements are basically designated by C. Okay, so most of complements which was earlier discovered, they were given the name as a C, and there is a there will be a numbers. Okay, and there is also some complement name is also as a trivial name. Okay, for example, factor D and factor B, something like that. Okay, so this is first step in complement. Okay, and generally when this complement get cleaved or proteolytically activated by previous complement, then this breaks into two fragments. Generally these fragments are one larger component and another is a smaller component. So the smaller component in general designated by A. Okay, so for example, there is a complement C3. Okay, and if C3 is activated, then it will make a two fragments, C3A, which is a smaller fragment, and there will be a C3B, which is a larger fragment. Okay, and this this naming is present in this whole complement system. You will see shortly when you will see the pathway, activation pathway of complement. Okay, the B part is always the larger fragment in general. Again, it's not always the case. In general, the B part is the larger fragment and A part is the smaller fragment. And so there is a larger fragment which we designate as a C3B, and this larger fragment is basically in various complement you will see there is a larger fragment and they are designated as a B. For example, if C3 will cleave, then it will make a smaller component, that is C3A, and the larger component C3B. Okay, and these complement upon cleavage and binding with other complement, they gain the enzymatic activity, and this enzymatic activity we basically denote by a bar. As you can see that here there is a C4B2A. So this C4B2A protein complex is basically interacted with C4B and 2A. Okay, complement C4B and complement C2A. This makes a complex, and when this complex is formed, then this will gain the enzymatic activity. Okay, and when the complex gain the enzymatic activity, this is denoted by bar. Okay, so now I will talk about the complement. Basically these complements are classified into 7 functional categories. Here you can see there are various functional category. The first category is basically initiator, the complement which initiate the complement fixation pathway. Okay, here you can see that there will be a C1Q. C1Q is a initiator, and later on you will see that this C1Q is an initiator of one particular pathway which we call it as a classical pathway. Okay, the complement molecules which activate another pathway, we are also categorize these proteins in as an initiator. For example, MBL, phycols, properdin or factor P. So these are all these molecules are basically initiator which will activate or initiate various complement pathway. Okay, there are 3 complement pathway which you will see soon after this slide. Okay. Another is activating enzyme. So there are complement which basically activate the enzymatic activity like C1R, C1S, C2A, CB, capital B, small b, factor D, MASP1, MASP2, MASP3. So these are basically activate the enzymes. Okay, they trigger the enzymes. Okay. Another is surface binding protein and opsonins. I have explained you what is opsonization. So all those molecule which is involved in opsonization, we call it as opsonins. So these surface binding protein and opsonins are C4b and C3b. So if microbe is coated by C4b or C3b, then it will be readily phagocytosed. Okay. So this is the one category. Another is peptide mediator of inflammation. I told you the complement breaks into 2 component and one of the component basically induces inflammation. Okay. So those component we call it as a peptide which basically induces inflammation, that is C5a, C3a and C4a. Okay. The outcome of complement activation, I told you the lysis. Okay, one of the outcome is lysis. Okay, so this lysis is basically mediated by one protein complex which we call it as a membrane attack complex. Okay, and all those complement which is involved in this making this complex, we call it as a membrane attack proteins. So all these molecules like C5b, C6, C7, C8 and C9, basically they make a constituent of this membrane attack complex. Basically it makes a kind of pore or hole in the target cell. Okay, this membrane attack proteins makes a pore or hole. Okay. Another is a complement receptor. So these complements, for example C4b and C3b, they after coating there will be a receptor on phagocytic cells. Okay. So those receptors are categorized as a complement receptor like CR1, CR2, CR3, CR4 and CRIG. Okay. So these are the complement receptors. There are regulator protein in this whole complement pathway. We call it as a complement regulatory protein, that is C1INH, C4BP and CR1, or it is alternative name is CD35, MCP or CD46, DAF or CD55, factor H, factor I, factor P and CD59. So these are the various regulatory protein, and I will discuss some of these protein when I will talk about complements and disease. Okay. Now this complement pathway are basically there are three major pathway or 3 pathways, not major, all these 3 pathways are existing. First is alternative pathway, and this alternative pathway is basically activate spontaneously when there is a microbial infection. Okay. So in this pathway basically C3, there is a complement C3 which basically spontaneously hydrolyze, okay, and initiate the depositation of this C3 convertase on microbial surface. So what is basically happening, this when there is a microbial infection, there are some factor over the microbes which is distinguishing feature between our own cell and microbial cells. Okay, I will discuss when I will take up the alternative pathway, but just you remember here that there are some distinguishing features which basically allow the initiation of hydrolysis of C3. C3 is a complement 3. So this initial hydrolysis basically cause the depositation of C3b over the target cell, and then this will interact with another factor, and then that will acquire the enzymatic activity. Okay. So alternative pathway is spontaneously triggered when there is a microbial infection. Okay. Another is the classical pathway, and this classical pathway is basically initiated, as you have seen in previous slide. There will be a molecule known as C1q. Okay. This C1q molecule, when interact with antigen antibody complex, then this complement initiated. So classical pathway is triggered by antigen antibody complex. Okay. And eventually towards next step, this will basically make a C3 convertase. Okay. And then the subsequent pathway will be triggered. Another pathway is lectin pathway. This lectin pathway, I have told you there are lectin proteins. So what is lectin? Lectin is basically protein molecule which is binding with the sugar molecule. Okay. So this lectin pathway is triggered by mannose binding lectin. Okay, MBL. I have told you in previous session when I was discussing the humoral component. Okay, MBL. So MBL can also activate the complement pathway, and this we call it as a lectin pathway. Okay. So this will be triggered by various molecules like MBL, ficolins. Okay. And then this will activated, and then it will initiate the complement pathway. Please remember, the aim of all these three complement pathway is to make C3 convertase. Please remember, again I am telling, the aim of all these pathway, alternative, classical, or this lectin pathway, is to make the C3 convertase. And what is C3 convertase? It will cleave the C3. Okay. Or this cleaved C3 will generate C3a and C3b. Okay. So this C3a will be a kind of anaphylatoxin, or it will induce the inflammation, and C3b will be deposited over the target cells. Okay. And once it will be deposited, then various the most important thing which will happen is the formation of membrane attack complex, which you will see in subsequent slide. So here you can see that all pathway basically generates C3 convertase. Okay, which cleave the C3 and leaving C3b over the target cell surface. Okay, and this C3b will be bound to the microbial surface and release C3a. So C3a will be causing inflammation, and C3b will be present on the microbial surface, and once it will be present over the microbial surface, it will be readily phagocytosed. Okay. And there will be a various thing will happen. For example, here the C3a and C5a recruit phagocytic cells. So it is an inflammatory molecule. Please remember, this is an inflammatory molecule. So this inflammation will attract the phagocytic cell, and this will once it will be attracted, then they will clear the microbial pathogen. Okay. And C3b, as I have explained you, C3b, if the microbial pathogen is coated with C3b, then it will be readily phagocytosed. we call it as the opsonization. Okay. Opsonization is a process of this coating of this target microbial pathogen by C3b. Okay. And after that, this will be phagocytose. The third outcome here you can see, which I have shown you in previous slide, that will be the formation of membrane attack complex, and this membrane attack complex will make a pore in the target cells. Here this is a little more complex and less interactive. Here you can see that there are alternative pathway, and this alternative pathway is basically initiated. If you remember the previous slide, this basically spontaneously there will be a hydrolysis of C3, and then this spontaneous hydrolysis will make a C3b, and then this will interact with factor, there will be a factor B. Okay. And this factor B will again cleave, and then that will make a BA, and there will be a small the factor B will, once it will be cleaved, it will make a factor B small a, which will be a kind of anaphylotoxin and inducing the inflammation. The bigger component, that is factor B, means capital B and small b, that will make a, that will interact with C3b, and this C3b will be the C3 convertase. Okay. In case of alternative pathway, in case of classical pathway, there will be a C1qR2S2, this will interact with antigen antibody, and then this will basically make a C3 convertase after cleavage of C4 complement and C2 complement, and eventually that result to the formation of C4b2A. This will have a C3 convertase activity. As you can see in this slide, there is a bar. Okay. Both classical and lectin pathway will generate this kind of C3 convertase. So I will discuss all these pathway in more detail when I will take up individual pathway. Okay. So with this I am stopping here, and next session we will discuss about the particular pathway. Thank you.